Okay, so this is part 2 in the continuation in the steps in fertilization. The third part is the fusion of the oocyte and the sperm cell membrane. So the plasma membrane of the oocyte and sperm fuse and break down at the area of fusion. The head and the tail of the sperm enter the cytoplasm of oocyte while the plasma membrane remains behind. Okay, and these are upon the entry of the spermatozoa onto the oocyte, the following event occurs. Okay, first is there is cortical granule reaction and zonal reactions. So the release of cortical granules, the oocyte become impermeable to other spermatozoa, and the zona pellucida alters its structure and composition to prevent sperm binding and penetration. So these reactions prevent polyspermy, or yung fertilization of more than one spermatozoa. Okay? So, the mechanism of polyspermy will be discussed later on after the steps in fertilization. Okay? And after the cortical granule reaction and zonal reactions, next is the resumption of the second meiotic division. Okay, so the oocyte completes its second meiotic division immediately after penetration of spermatozoon. So if you can remember, okay, the development of the oocyte is arrested at prophase of meiosis 1. Okay, so it continues, it resumes its meiotic division when it is penetrated by its spermatozoon. Okay, and after that, there is the metabolic activation of the egg. So after the penetration of the sperm, it results to rapid intensification of egg's respiration and metabolism. Okay, the rise in calcium ions in the cytosol increases the rates of cellular respiration and protein synthesis by the egg cell. Okay, and with these rapid changes in metabolism, the egg is said to be activated. Okay, and the spermatozoon moves forward until it lies close to the female nucleus, and its nucleus becomes swollen and forms the male pronucleus. The tail detaches and degenerates. Okay, so ayan na, nag-fuse na siya. Okay, so these are the mechanisms to prevent polyspermy, or the fertilization by more than one spermatozoon, and it is observed by the eggs of the sea urchin and the oocytes of sea urchins. Okay, so we have the fast block to polyspermy. The fast block to polyspermy involves a change in electric potential of the egg plasma membrane. So within 1 to 3 seconds after binding or penetration of the sperm, the membrane potential shifts from negative 70 millivolts to positive 10 millivolts. Thus, hindi na pwedeng makapasok yung ibang mga sperms. Okay? And this slow block to polyspermy, on the other hand, is due to the mobilization of calcium ions from store bound within the egg. So, a wave of calcium ions propagates from the point of sperm entry kung saan nag-penetrate yung first sperm. And it causes cortical granule to fuse with the egg cell membrane. And at the point of entry, it releases contents of the granules cause the vitelline membrane to rise and harden into the fertilization envelope. That's why hindi na siya ma-penetrate ng other sperm cells. Okay? So what are the results of fertilization? So first, it stimulates the secondary oocyte to complete meiosis. Okay? And it restores the normal diploid number of chromosomes. So yung mga gamits natin, we know that they have haploid number of chromosomes due to the meiotic division. And with fertilization, it restores the normal diploid number of chromosomes, which is 46 in humans. Okay? And it results in variation of human species as maternal and paternal chromosomes intermingle. Okay? So, yung genetic information of the father from the sperm cell fuses with the genetic information of the mother from the oocyte, at nagkakaroon ng variation. Okay? Nakakahalo yung chromosomes nila. Okay? And the embryo contains only maternal mitochondria because the sperm mitochondria are dispersed into the egg cytoplasm and discarded. So, doon sa may third process kanina o events, doon sa penetration ng sperm and egg, kita natin nag-fuse yung membrane, sa membrane ng egg, ng oocyte. Okay? Yung tail and yung cytoplasm. Okay, so at the end ay discard din or nag-degenerate din yung tail. So nawala yung mitochondria ng sperm cell. Okay, and last, it determines the sex of the embryo. The sex chromosome Y or X carried by the successful sperm determines the embryonic sex. Okay, so let's now move on sa cleavage. Okay, so eggs, we know that this is the female gamete. Okay, these are round or oval reproductive body consisting usually of an embryo surrounded by nutrient material and a protective covering. Okay, and these eggs, okay, kapag na-fertilize, will have polarity. The eggs and zygotes of many animals have a definite polarity. So this polarity is defined by the distribution of the yolk. Okay, so we have two poles, the vegetal pole which has the most yolk and the animal pole which has the least yolk. Okay, so ito yung example. Okay, the animal hemisphere or the animal pole, yung kukunti ang yolk, at si vegetal hemisphere or vegetal pole yung may mas madaming yolk. So these are the classification of eggs according to the amount of the yolk that they have. Okay, so first we have alecytal eggs. Alecytal eggs. Okay, these are those without or with a very small amount of yolk. Okay, so for example, yung humans. Okay, kasi hindi naman natin kailangan ng yolk. Okay, then next is we have the mesolecytal or microlecytal. So these eggs contain small amount of yolk such as those of the Amphioxus. Okay, Amphioxus. Then we have mesolecytal eggs. These eggs have moderate amount of yolk as in frog and toads. Okay, and sa fourth, we have the polylecytal or megalecytal eggs. They contain large amount of yolks as in fishes, reptiles, and birds, because solely they depend their nutrition on the yolk as they mature or develop inside egg. Okay, so this classification naman is according to the distribution of yolk. Kanina dun sa amount ng yolk meron ang eggs. But now, we will discuss kung ano yung different classifications of eggs according to the distribution of yolk. So we have the isolecytal egg, where the yolk is evenly distributed. So examples are mammals. Okay? Ayan. So isolecytal, so all of the cells are have equal amounts of yolk. Okay? And we have the telolecytal. Okay, yolk is unevenly distributed and usually concentrated on one side of the egg. So the examples are fishes, amphibians, and reptile. So we have this type of eggs, okay, sa telolecytal. Okay, and we have the centrolecytal. Okay, from the root word or first word is centro. Ibig sabihin, the yolk is concentrated in the center. So eggs of insects are of this type, kung saan nasa gitna lang yung yolk nila. Okay, so makikita nyo yung mga dots dyan, those are yan yung mga nagdi-divide na cells. Okay, so yung nasa gitna yung yolk. Okay, in terms of cleavage, ito yung form kapag nagdi-divide ang ating zygote. Okay, so we have different types of cleavage. Okay, we have the first holoblastic cleavage. So the fertilized egg divides completely into two separate cells. Then each cell divides completely again to produce 4 cells and again and again. So exponentially yan na nagdi-divide. Okay, cell division, typical cell division mitosis. Okay, from 1 cell to 2 cell to 4 cell to 8 cell to 16 cell and so on and so forth. So we have two types of holoblastic cleavage. So we have the total and equal. So this type of cleavage is when segments are of equal sizes, as in echinoderms, amphioxus, and animals. Okay? Ayan yung example ng holoblastic cleavage. So, same sizes yung mga cells kapag sila ay nagdi-divide. And another type is the total and unequal. So, this is the condition when the segments on the animal pole are smaller than those on the vegetal pole, just like in fishes and amphibians. Okay? So, sa unang mga stages, in the 2 cell to 4 cell stages ay sabi natin pare-pareho, but later on, makikita natin that the animal pole is smaller than the vegetal pole, or the cells in the animal pole is smaller than the cells dividing in the vegetal pole. So, that it is total and unequal. So, meron din naman tayong tinatawag na meroblastic cleavage. So, this is the partitioning of the cell is incomplete and confined to a small disk of the yolk-free cytoplasm, as in the eggs of the birds. So, in the laboratory, we will take a look at kung ano ang nangyayari dyan sa partitioning ng cell doon sa small disk na yun. So, meron lang part doon kung saan nagde-divide ang cell. And the rest is the yolk. Okay? That is meroblastic cleavage. And the third one is the superficial cleavage. Okay? So, when in early stages, cleavage is incomplete and only the nucleus divides. Okay, later, the daughter nuclei migrate into the peripheral cytoplasm and form a single layer of cells at the surface. And this is observed in centrolecital eggs of insects. So, nakita natin kanina, meron lang isa na cell, then nag-divide siya at napunta siya doon sa peripheral o sa paligid at naiiwan sa gitna yung yolk. So, this is the example. So, this is the cell, yung dot na yan. Makita natin, it divides into 4. Okay, 4 cell stage, then so on so forth hanggang sa mabuo doon sa paligid o sa peripheral, sa paligid noong developing embryo. Okay? So, as we talk about cleavage, we can also discuss the different types of twins. Okay? There are 3 types of twins. There are monozygotic o yung identical, dizygotic or yung fraternal, and conjoined or Siamese. So, let's first discuss ano ba yung monozygotic or identical twins. So, from the term itself, mono, ibig sabihin monozygotic, from one zygote or one fertilized egg lang na derive yung dalawang individuals. Okay? They arise from subdivision or splitting of a single embryo. Okay? Kaya mostly magkakamukha ito. So, ito yung mga examples ng mga monozygotic twins. Okay? So, sino nakakilala sa ano na? Richard and Raymond Gutierrez, George and Jai, si ano nang pangalan nito ba na? Sa PBB din. And international vloggers, the Dobre brothers and the Stoke twins. Okay. So, makikita nyo na sa yung pagkakahawig, pagkakamukha, but there are still features na hindi sila magkapareho. Okay? And the second type of twins are the dizygotic or fraternal twins and are the product of fertilization of 2 ovulated ova. Okay? So, ito yung masipag maglabas ng eggs. Okay? Kaya dalawa ang eggs na nailabas at dalawa ang na-fertilize. Okay? So, ayan. sila the Legazpi twins. Okay, tama ba? Okay. Villarroel pala. Tama? Legazpi. Sorry, Legazpi ako po. So, the Legazpi twins are examples. Okay. And these are conjoined twins naman, or yung Siamese twins, are result of partial separation of fertilized egg. Okay. So, ito yung hindi fully nag-separate. So, there are cases na pwede naman silang paghiwalay yung pagtanda or pagkalabas nila, but there are some na mahirap because some of them may share only one heart, okay, one organ, at wala dun sa kabila. Okay? So, as part of the discussion of their cleavage, okay, nagde-divide ang cells natin. So, we have the different stages of embryonic development, and after many divisions of the cell of the embryo, we will come to a point na makakakuha tayo ng hollow ball of cells with a cavity, or what we call the blastula, in the process of blastulation. Okay? So, this blastula is the single layer of hollow ball of cells with a cavity. Ibig sabihin, sa loob walang laman. Okay? And the cavity that forms with the blastula is called the blastocele. Okay? Yung cavity na form sa loob is yung blastocele. And ang tawag natin sa blastula ng mammals ay blastocyst. Okay? And it consists of inner cell mass, which will form the embryo, and outer layer of cells, the trophoblast, which will develop into the placenta. Okay? So, ayan. It is a hollow ball of cells. Okay? Titignan natin sa labas dito sa figure A. Okay? And sa figure B, ito yung cross-section ng ating blastula or blastocyst. Okay? So, we have the layer of cells, single layer of cells, and yung sa loob ay wala. Sa cavity, okay? Walang laman. Okay, this is the blastocele. And to view the trophoblast, ito yung sa mammalian. So, this is a blastocyst. Okay? So, we have the single layer of cells outside, which is the trophoblast. Okay? And we have the inner cell mass. And we have the blastocele. Okay? So, itong inner cell mass siya yung magpo-form na ating embryo. Okay? And yung trophoblast, yung develop na placenta. Okay? So, after blastulation, we will, the embryo will undergo gastrulation. So, gastrulation is the process of morphogenetic movements of the cells that transform the plastura into a three-layered embryo. So, hindi naman mag-stay na ganun lang, single layer ang ating embryo. Okay? So, under gastrulation, ito na yung movement ng isang layer ng embryo na yun. In further division of the cells of our embryo. So, during gastrulation, cell movements result in massive reorganization of the embryo from a simple spherical ball of cells, yung blastula, into a multi-layered organism. So, sa blastula or blastocyst, meron lang tayong single layer of cells. However, during gastrulation, nagkakaroon tayo ng different layers ng cells. So, it consists of 3 germ layers: the outer, the ectoderm, the middle, the mesoderm, and the inner, pinakaloob, is the endoderm. Okay? So, the common feature of early gastrula is blastopore, an indentation or groove through which cells move inside to form the endoderm and mesoderm. And the fate of the blastopore is used as the basis of classifying the animals into 2 groups. Okay? So, makikita natin yung movements, morphogenetic movements mamaya sa gastrulation at makita natin yung blastopore na sinasabi dito. Okay? So, yung dalawang groups ng animals are the protostomes and deuterostomes. So, protostomes, the blastopore develops into the mouth. Okay? Yung deuterostomes naman, the blastopore becomes the anus and mouth formation is secondary. Okay? So, mauuna muna ang anus and then susunod ang mouth. Sa protostomes naman, mouth lang ang made-develop sa blastopore. Okay? So, the first morphogenetic movement under gastrulation is invagination. Okay? So, remember, yung ating blastocyst or yung blastula, yung single layer na yun, ang mangyayari, ayan, magkakaroon ng invagination or the infolding of a sheet of a cell. So, in a portion of the cell, okay, magkakaroon ng invagination. Parang papasok pa loob yung ating cells. And at some species, meron ding evagination or the outpacketing of a sheet of cells. And after invagination, we have the involution. Okay, is that interning or inward movement of an expanding outer layer so that it spreads over the internal surface of the remaining external cells. So an epithelial sheet rolls inward to form an underlying wait lang. Matakpan, hindi ko na-edit. Okay, underlying layer. Okay? So, ito na yung product ng ating invagination. So, nakita nyo yung color, yellow, and na-coat na niya yung buong ano. Okay. Tapos, dito, doon sa may part ng invagination, ayan, dyan naman yung mangyayari yung involution. Okay? So, paloob, inward yung movement para magkaroon tayo ng another layer ng cells. And after that, yung ingression. Okay? It is the sinking of individual cells from the surface layer into an area. So, this time, may migration na nangyayari. So, individual cells live on epithelial sheet and become freely migrating mesenchyme. So, after ng involution, okay, nabuo na natin ang 3 layers. So, magkakaroon naman tayo ng ingression wherein some of the cells will leave yung epithelial sheet. Okay, they migrate into other areas of the developing embryo. And next is the delamination, or the splitting of one cellular sheet into 2 or more parallel sheets. And after that, we have the epiboly, or the spreading of a cell sheet. So those are the morphogenetic movements involved in gastrulation in order for the cells to form yung 3 layers natin. The ectoderm, the mesoderm, and the endoderm. And after gastrulation, we have neurulation. Okay? With the term neuro and neurulation, alam natin that it is the formation ng ating it has something to do with the formation of the central nervous system. Okay? And it is the process involved in the formation of the neural tube, which gives rise to both the spinal cord and the brain. Okay? So, the neurula is the stage in which the embryo develops the rudiments of the central nervous system. Okay? And as neurulation continues, the central nervous system appears sequentially in the form of neural plate, neural groove, and neural tube. Okay? So, we will explain more of this sa may laboratory part na lang din natin para makita natin yung different areas ng central rudiments ng central nervous system, with the neural plate, neural groove, and the neural tube, kung paano nangyayari na bubuo ang ating central nervous system during neurulation. Okay? So, under neurulation, the ectodermal cells flatten into a neural plate. Okay? And the center of the plate sinks, forming neural groove. And the edge of the plate is elevated to form neural folds. At yung neural folds, mag-fuse and form the neural tube. Okay, so the anterior end develops into brain and the posterior end develops into the spinal cord. So after the neurulation, okay, or the formation of the rudiments of the central nervous system, we will have the organogenesis, or the formation of tissues and organs, okay, to form the from the 3 germ layers nung nasa gastrulation. Okay, so the cells of the germ layer divide into smaller groups that develop into organs of the individuals. So this is known as the primary organ rudiments. So from the primary organ rudiments, the secondary organ rudiments develop. Okay, and organogenesis begins with the development of the nervous system. So mauna munang ma-develop ang nervous system after noong mga susunod na mga organ system. Okay? And the ectoderm, these are the fate of the cells in the ectoderm. Okay? They will form the epidermis of the skin and the skin derivatives like skin glands and nails, the epithelial lining of the mouth and rectum, okay, the nervous system and sensory structures, and the pineal and pituitary glands. So remember, these are the fate of the cells from the ectoderm. And moving on sa mesoderm, ito naman yung mga mafu-form na organs from the mesoderm. The notochord, the skeleton, the muscular system, the circulatory system, the excretory system, reproductive system, peritoneum, adrenal cortex, and the dermis. Okay? And dun sa inner parts ng cell, which is the endoderm, ito naman yung fates ng cells na mabubuo nun. The digestive tract lining, the respiratory system lining, most of the endocrine glands, liver, and the lining of reproductive and excretory system. So those are the different ontogenetic processes, okay, from the hollow ball of cells, the vasculation, up until the organogenesis. So these are the events that will happen after the fertilization of the oocyte from the penetration of a sperm cell. So that's it for the topic under the fertilization and different ontogenetic phases of development.